Our journal looks to determine the effect of insulin deprivation in human Sertoli cells on their metabolism and metabolism-associated gene expression.
The main techniques used are proton nuclear magnetic resonance and reverse transcriptase polymerase chain reaction.
The mRNA expression level of glucose transporters GLUT1 and GLUT3 were determined by RT-PCR.
The absence and presence of insulin and metabolite variations were determined by proton NMR.
Sertoli cells have a high capacity to produce lactate and preferably use lactate for the energy metabolism instead of glucose.
Human Sertoli cells are usually referred to as nurse cells for the reason that they supply physical and nutritional support for germ cells.
Germ cells give rise to gametes due to the process of meiosis.
Sertoli cells are found in the convoluted seminephros tubule, which is located in the human testicle.
They metabolize glucose into lactate to nourish sperm cells during spermatogenesis.
Spermatogenesis is the process by which male germ cells, called spermatogonia, undergo meiosis and produce a number of cells termed spermatozoa, which are also known as sperm cells.
Regulation of glucose metabolism in human Sertoli cells is critical for fertility and spermatogenesis.
Insulin regulates glucose uptake.
Human Sertoli cell metabolism and metabolism-associated gene expression are negatively affected by the deprivation of insulin.
It is this insulin deprivation that influences major regulatory systems, and the deprivation of it results in many problems such as diabetes mellitus, hypertension, and others. Reverse transcriptase polymerase chain reaction is a process in which artificial DNA is replicated and used on small samples of DNA to generate multiple copies. It's a variant of PCR and is commonly used in molecular biology to detect RNA expression levels. Reverse transcriptase PCR creates complementary DNA transcripts from RNA in order to quantitatively detect gene expression. It's useful for detecting cell-specific gene expression shown by the presence of specific mRNA when the amount of biological material is limited.
The two complementary strands of double-stranded DNA are usually differentiated as the sense strand and the antisense strand. The DNA sense strand looks at the messenger RNA and can be used to read the expected protein code. The DNA antisense strand serves as the source of the protein code. Due to the fact that its base is complementary to the DNA strand, it is used as a template for the mRNA.
Reverse transcriptase PCR relies on the following facts. DNA is made up of an antiparallel backbone, meaning that the sugar-phosphate backbone run in opposite directions. Also, that they have strands with 5' and 3' ends. DNA grows from only the 3' ends, and that there is complementary base parents.
Using either oligo-DT primer to anneal to the 3' polyadenile tail of the mRNA, or using random hexamer primers together with reverse transcriptase, complementary DNA, otherwise known as cDNA, is produced, which is then amplified by PCR.
RT-PCR in this experiment determines mRNA expression levels of glucose transporters GLUT1 and GLUT3.
In order to determine the effects of insulin in metabolite consumption and production, as well as GLUT1 and GLUT3 expression, Sertoli cell medium was supplemented either with insulin transferrin sodium selenate, which is the control group, or with transferrin sodium selenate, which is the insulin-deprived group.
Treatments were performed for 48 hours. Various conditions were used for incubation, and sodium fumarate of a concentration of 2 millimoles was used as an internal reference at 6.5 parts per million in order to quantify the level of the metabolites in the solution.
It was a 48-hour cell incubation process, and during that time 250 microliters of the culture medium were collected at 6, 24th and 48-hour intervals for proton NMR analysis. At the end, cells were detached from the flask using a trypsin EDTA solution and collected for RNA extraction.
The table shows primer sequences, optimal annealing temperature, which is 80 in the table. C in the table shows number of cycles required for exponential amplification phase of fragments. Amplicon size, which is size of piece of DNA formed from amplification events, are all indicated in table 1.
Proton NMR spectroscopy investigates the absorption of radiation by a nuclei of hydrogen atoms and is therefore a technique of obtaining information about the number and arrangement of hydrogen atoms in a molecule. Proton NMR is used in order to determine the metabolite variations in the absence and presence of insulin in human Sertoli cell-enriched cultures.
Protein NMR was used to determine lactate production, glucose consumption, and variations in substances such as pyruvate and alanine.
In the first 6 hours of the treatment, the glucose use was almost zero for insulin-deprived cells. The cells in controlled conditions consumed 28 plus or minus 3 picomoles of glucose. The total consumption of glucose after 48 hours of treatment did not differ much in the cells from both groups.
In figure A and B, between the 6th and 24th hour, the insulin-deprived cells show a high glucose consumption rate. This shows that glucose metabolism was favored in an environment where insulin was absent. At the end of the 24th hour, the human Sertoli cells cultured without the insulin consumed 50 plus or minus 6 picomole per cell.
Insulin deprivation decreases lactate production. Amount of lactate produced in the first 6 hours of treatment was similar for both control and insulin-deprived cells. Throughout the next 42 hours, human Sertoli cells in control conditions continued to make lactate at a similar rate. However, insulin-deprived cells secreted significantly lower levels of lactate. At the end of the 48 hours, human Sertoli cells from the control group produced 12.5 plus minus 0.3 picomole per cell. An insulin-deprived group produced 4.1 plus minus 0.2 picomole per cell.
Insulin deprivation decreases GLUT3 and increases GLUT1 mRNA. GLUT1 and GLUT3 are high-affinity glucose transporters on plasma membranes of Sertoli cells and are associated with the transport of glucose in cells with high energy demands.
The mechanisms that regulate Sertoli cell glucose metabolism are relevant in maintaining spermatogenesis and fertility. Insulin-deprived human Sertoli cells consume significantly less glucose and pyruvate during the first 6-hour period.
Glycogenolysis pathway may also be participant in this process, as glycogen and glycogen phosphorylase activity have been detected in Sertoli cells, making possible that glycogen hydrolysis fuels glycolytic pathway in the cells.
Sertoli cells adapt their glucose uptake mechanisms as indicated by GLUT1 and GLUT3 mRNA modulation in order to maintain the lactate production within a physiological range.
After 48 hours, the glucose consumption was not significantly different between human Sertoli cells from both groups, although lactate production was significantly lower in the insulin-deprived cells.
Sertoli cells can take up glucose from the external medium through the action of GLUT1 and GLUT3, which are present on the plasma membrane. This action is a rate-limiting step in glucose metabolism in human Sertoli cells.
After the 48-hour insulin deprivation treatment, there was a significant decrease in GLUT3 and an increase in GLUT1 mRNA levels.
These changes are not a direct measure of protein function, but they show the effect on the regulatory mechanism action of these proteins by insulin. Therefore, insulin-deprived human Sertoli cells adjust their glucose uptake by modulating GLUT1 and GLUT3 transcript levels.